Dear learners, welcome back to NPTEL online certification course on novel drug delivery systems. I am Dr. Satish Devanapelli, Assistant Professor, Department of Pharmaceutical Sciences and Technology, Institute of Chemical Technology, Mumbai. So, in our novel drug delivery systems, we are discussing on ocular drug delivery systems. In that, we have started with the novel ocular drug delivery systems. In our previous session, we have discussed the what are the in situ gelling systems, how that can overcome the conventional solutions. Then after that, we discuss second topic, that is the contact lenses. We have seen the properties of the contact lenses, how that will be different from the conventional systems, and we have seen the advantages. And also we have discussed the importance of drug coated contact lenses for the treatment of eye diseases. And the last, we have discussed ocular inserts, and again it is further classified into the insoluble ocular inserts and soluble and then erodible. So, today we are going to discuss on the remaining topics. So, these four we will see, that is particulate systems. Again it classified as a nanoparticles, microparticles, microemulsions, liposomes, niosomes and dendrimers. And then we will see in the microneedles, again we have various types of microneedles. We have nanowafers and then iontophoresis systems. So, in our week 2 and week 3, we have dedicatedly discussed nanoparticle as in drug delivery systems. So, there we have discussed very detailed on nano carriers. So, just for the refreshment, so we have discussed there on the polymeric nano carriers, and also we have seen the various types of the lipid nano carrier systems, and also we have seen some type of the vesicular drug delivery systems, and also we have discussed the some of the inorganic nano carriers. So, these we have classified based on the materials. So, today we are not going to discuss details of what are the carriers, but we will discuss for the eye disease treatment. So, how these formulations are different when compared with the conventional formulations. As we know, the nanoparticles are microparticles, so which the name given based on their size. So, which are solid in nature and which are multi-compartment polymeric drug delivery systems for the eye applications. Now, you can see the size range. These are the theoretical size range from 10 nanometer to 1000 nanometer. So, that is the size for the nanoparticles, whereas microparticles, so 1 micron to 5 to 10 micrometer is preferable for the applications. So, when you convert your conventional formulation into the nanoparticles, you will see few type of the advantages. Here I have given very limited advantages, but you can refer our week 2 and week 3 nano carriers as in a drug delivery systems. So, there we have discussed advantages as well as limitations of all the carriers in each class of the polymer, lipid and inorganic and vesicular drug delivery systems. So, the major advantage is that the improved corneal penetration. Now, in our first class we discussed the barriers are the one of the major challenge in the ocular drug delivery. If you would like to overcome the absorption or penetration properties, so these nanoparticles are the very tiny particles and these have ability to cross all type of the barriers which are present in the body at the cellular levels. So, and when you convert your drug solutions into the nano form, so definitely you will see the improvement in the corneal penetration. And not only that, when you reduce the particle size, your surface area will increase, that ultimately improve the dissolution, nothing but solubility, and that leads to the enhancement in the bioavailability of your drug. So, with the nano system we can expect the increased bioavailability as well as with the help of the solubility improvement and as well as the permeation improvement. So, yes, the problem here is low drug loading capacity when compared with the convention. Yes, here you can use when your drug is potent, and such cases you can go ahead with the nano carrier systems. So, these are the drugs we can people have converted into the nano carrier systems for the treatment of various eye diseases. Now, the second category is the microemulsions. Now, the definition says that microemulsions are thermodynamically stable, isotropic mixture of oil, water, surfactant. Again, we need to give co-surfactant also. So, that will form an emulsion which is not similar to the conventional emulsions. So, this is stable at thermodynamical conditions, and this is very clear solution, and when you see the particle size and you will get the size range in the nanoscale because of the great advantage of these combinations of surfactant, co-surfactant have the high reduction in the interfacial tension compared with the conventional emulsions. Here you no need to use any type of high energy to convert into the nano emulsions. So, these are the instant will get instantly when you add this oil, water, surfactant with your drug, and it will give the size range the below 100 nanometer. So, typically sometime we will observe even 10 nanometers also. So, these are very teeny nano drops and their permeation is also very high. They will stick to the corneal surface and it can also act as a drug reservoir compared with the conventional solutions, which will also reduce the tear fluid and also drug loss. One more interesting thing that here we can use some of the mucoadhesive materials on with this microemulsion. So, that will also helpful as in a bioadhesive systems. So, these are the advantages. We can improve the bioavailability, also we can enhance the contact time with the eye. We can also, these are the proven clinical studies on the healthy voluntaries, which will help in the improvement in the bioavailability. So, these are the few drugs which we can convert. So, here we use most of the cases water insoluble drugs we can convert into the solubilized format by using the simple microemulsion strategy. And not only that, these emulsions without oil you can also convert into the conventional formulations. You can convert into the again the semi solids and again into the solids, that is nothing but self micro emulsifying drug delivery systems. With that you can improve the stability of your formulations. Now, the third category, this all we have discussed in our nano carriers, just for the we are seeing the ideas of these nano carriers for the ocular drug delivery systems. So, the third one is liposomes, which are the self assembled bilayer phospholipid vesicles, which are microscopic and the spherical in nature, that can encapsulate both hydrophilic as well as hydrophobic substances. That is the beauty of the liposomes. These are prepared by the phospholipids, which are biocompatible as well as biodegradable, non-toxic. So, with that we can use both type of drugs, water soluble and water insoluble drugs, and the permeation is very high compared with the polymeric carrier systems. This will also protect the drug from the enzymatical degradation on the corneal surfaces. Yes, the phospholipid is very less stable compared with the polymers. It undergo rancidity, oxidation. So, for that we need to provide antioxidants, and the loading also very less. If it is a water insoluble drug, the chance of loading will be very high. And then the scale up also one of the major issue, and the very expensive, and the establishment of the regulatory and other technological, the guidelines are not yet established for some of the formulations. So, that need to be considered while developing the formulation. So, these are the drugs we can make liposomes for the treatment of the eye diseases. The Niosomes are the non-ionic surfactants which are alternative to the liposomes, because liposomes are not stable in the various storage condition as well as in the formulation with the drugs also. So, these are the non-ionic surfactants are the alternatives of the liposomes, which again form the vesicles, and as we know that non-ionic surfactants are not much toxic compared with the cationic and anionic surfactants. So, here also we can load both type of the drugs, the hydrophilics and hydrophobic drugs, and it will form the bilayer structures. These are stable compared with the liposomes, and yes, some of the disadvantages of the which will avoid the disadvantage of the liposomes, that is chemical instability, oxide degradation, can be overcome by using the Niosomes, and which will increase the drug contact time with the cornea, and it will improve the bioavailability of the Niosomes. Now, here the another one is dendrimers. As we know that you can see in the structures, the dendrimers are the branched spherical monodisperse 3D polymer structures. Now, it looks like a tree which contain the core and then branches and then terminal functional groups. So, terminal functional groups help for the functionalization. There we can modify the surface, you can see here in the picture. So, this will help for the further modification with various types of the conjugation with the lipids and other active molecules. So, with this we will make the dendrimers and there we can load the drugs. So, that will help for the permeation as well as to control the release. So, you can see the sustained release and also it will help for the treatment and that can be placed, even it will help for the antibacterial and corneal tissue engineering. But we need to consider the cost of the total formulation is very important. All drugs cannot be converted into the nano carriers. If the cost of the drug is very less, very cheap, and you cannot go for the control drug delivery, or you can say the nanoparticulate system, because of the polymer itself is very costly. Yes, very needy in the treatment and it is a challenging. So, such cases we can convert into the nano carriers. Now, here we are not going to discuss all type of particulate systems. So, with this summary we are going to conclude the particulate systems. So, here you can see various types of the carrier systems. All this we have discussed in week 2 and week 3. So, here is the solid lipid nanoparticles, polymer nanoparticles we can use for the treatment of sclera. Now, for the retinal treatment, SLN, liposomes, micelles, nanomicelles, nanoemulsion. Now, optic nerve again albumin particles, chitosan, polymeric, and for choroid dendrimers can be used, iris, polymer, and again for the cornea SLN, chitosan and the polymer. So, all this formulation can be possible and that need to be given with the right route also, right. Some of them anterior routes which may be topicals, injection, some of the intravitreal and also for the posterior segment. So, this need to be translated into right dosage form and that need to be delivered by right routes of administration, then only your treatment can be fulfilled. Now, the another topic that we are discussing on ophthalmic microneedles. So, this invention came from the topical transdermal patches. These are the needles which contain the micron size needles. You can say it is a combination of injections and the patches which are nothing but micro needles. You can see there are the needles on the patch. So, micro needles, so which are presented on the solid support, that is the patch, with the needle heights ranging between 25 to 200 micrometer. These are arranged in the patch. So, hundreds of microneedles are arranged in an array. Now, you can see here these are arranged here and also you can see here and also see here also in the surface morphology by using SCM. Okay. So, with this we will understand that it is the patch which contains the needles. These people use for the alternative of the transdermal patches. Now, most of the scientists are working and translating their conventional ophthalmic eye dosages, drugs for the treatment of eye disease are they are translating into the microneedles to have a better treatment. So, to deliver the sufficient amount of drug to give a required therapeutic response. Now, the beauty of this formulation is that which are the non-invasive methods which will deliver the drug into the eye. So, you can completely avoid the injections which are given into the eye at the anterior segment, intravitreal and the posterior segment that can be avoided. With that we can improve the patient compliance, also we can improve the therapeutic outcome compared with the traditional injection. Now, you can see the needles which can be prepared by using the polymers and other materials. So, these are the classification of the ophthalmic microneedles. So, here is the picture represented, it speaks about the solids, coated one, hollow, dissolving and the hydrogel. By seeing the mechanism you will understand, right, after administration. So, these are the solid, here we use the solid needle, there we will press on the skin and then we will put the patch. So, this is some type of the conventional type of the microneedles. Then after putting of your patch, then the drug will release. So, the here we use only solid metal type of the or polymeric type of the needles we use just to open the stratum corneum. Now, the second one is the coated one, same type of the needles, but which are coated with the polymers. So, what we here we do, we will administer on the skin, then we will remove the solid patch and then your coated material will be placed in the skin only, then later then it will take the water from the skin and it will release the drug, that is the coated one. And the hollow is nothing but, so here you can see, so it is a higher version of the coated one. Now, you can see here, here is your drug which is available in the microneedle itself, but here you can see there is a hollow, you do not see any coating on these needles. But after administration, so the drug will slowly release into the skin or into the administered site. So, that is called as hollow. So, it will absorb the water and then it will release the drug into the target site. And the fourth one is dissolving, that means it will completely dissolve or you can say disintegrate within the targeted site. And the fifth one is very advanced one, that is a hydrogel forming microneedles. Now, you can see here is the reservoir, their drug is loaded. Now, see how it is working. So, this polymer have high capacity to hold the water after administration. You can read here, drug molecules diffuse through the swollen microneedle. So, these are the materials, they have high water holding and swelling capacity, so that it will absorb the water and it will swells, and this release is the controlling release is will be very high compared to all other systems. So, it will works as a reservoir and the drug will release or diffuse slowly into the targeted site. So, these are the total 5 different types of the microneedles. Now, in eye, if you want to target the different sites, so the length of the needle need to be considered. Now, if you want to target a sclera, so here you can see with the pictorial representation, the 100 micrometer is sufficient for the treatment. Now, when you want to go for the choroid, so now you need 300 micrometer length of the needle. Now, if you want to go further deeper into the retina, you need to come up with the 500 micrometer and very sharp and the very lengthy type of the needles that will help for the treatment of the posterior eye diseases. So, these are the various types of the microneedles we can use for the treatment. Now, the another topic is ophthalmic nanowafers. So, these are like, say, you can say it looks like a tiny films, but which comes in the disc shape. So, that need to be placed on the eye, that will, after application administration, it will take the water from the tear fluid and then it will slowly release the drug. That is second step, it follows the dissolution. Then after that your drug will distribute within the eye region, and then after once the drug is entered into the blood and finish the treatment, and that will go away because of the biodegradation property or disintegration property of the materials. So, these are the nanowafers, or also will be considered as it looks like a nanofilms, which are coated with the drugs. You can see here in the pictorial representation. So, these are the wafers which will slowly release the drug into the eye. Another topic, that is ocular iontophoresis. Now, this is also considered as a non-invasive techniques. So, the microneedles also comes from the transdermal drug delivery system. Again, ocular iontophoresis also comes from the transdermal patches. So, here we use, definitely the drug should has the charge in nature. So, the drugs, the positively charged drugs are delivered from the anode. Now, you can see here, this is the anode. Here when you want to deliver the positive drugs, when you apply the iontophoresis, the anode will repel your positive molecules to the cathode. So, similarly same thing happens when you want to deliver the drugs which are in the negative nature, and you need to provide the cathode that will repel negative ions to the target site. So, by doing this, by applying of the iontophoresis, we can deliver the drugs. We can also overcome the barriers which are not possible by the topical administration. So, it offers fast, safe, painless delivery, and yes, we can also achieve high drug concentration at the target site. So, the drugs studied, which are gentamicin, dexamethasone, ciprofloxacin and ketoconazole drugs, can be used for the treatment of the eye diseases by using the ocular iontophoresis. So, with one picture representation we will understand how this works. So, here you can put the treatment. You can see here, these are the two anode and cathodes, okay, and that need to be placed on the eye, and then you need to start off the generator that will release the drug and that will reach to the target site in the eye. So, wherever the requirement, and that need to be fulfilled by using of the both positive and negative charges of the drugs by using the ocular iontophoresis. So, if you want to know more on this, you can prefer this article. So, that will help for understanding and other applications of the ocular iontophoresis. And the last one is filter paper strips. So, here these we use for the diagnosis especially, and when you want to know the infection status. So, such cases we can use filter paper strips, or it will looks like a films that need to be applied on the eye, and later that need to be removed, and then we need to quantify, or we can see the damage of the cornea, conjunctiva and eyelids, and also we can detect the number of microorganism present at particular site, and type of the microorganisms can be detected by using the various instruments. So, these are the filter papers also we can use for the detection and also for the diagnosis of the various types of the eye diseases and eye infections. So far we have discussed on the various types of the novel ocular drug delivery systems, and today we have discussed on the particulate systems, and also we have discussed the microneedles, and also we have seen what are the nanowafers also, and we have seen various types of other nano carrier systems. And in our upcoming classes we are going to discuss on the evaluation of ophthalmic formulations. So, with this we will stop today's lecture. Thank you so much.